La fundación de la Academia Nacional de Ciencias se debió a un grupo distinguido de peruanos dedicados a la ciencia, la mayoría profesores universitarios. Después de varios acuerdos preparatorios, el 6 de agosto de 1938, en el Salón de Actos de la Facultad de Ciencias Biológicas, Físicas y Matemáticas de la Universidad Nacional Mayor de San Marcos, se realizó la sesión de instalación de la Academia de Ciencias Exactas, Físicas y Naturales de Lima, acordándose su fundación y eligiendo a su primera junta directiva. El 23 de octubre de 1939, mediante decreto supremo firmado por el presidente Óscar Benavides, se hizo oficial la fundación de la Academia, bajo el nombre de Academia Nacional de Ciencias Exactas, Física y Naturales de Lima, con independencia en su organización y funciones. Años después, el 2 de diciembre de 1966, el presidente Fernando Belaúnde firmó un decreto supremo cambiando el nombre a Academia Nacional de Ciencias, como se le conoce hasta el presente. La Academia Nacional de Ciencias tiene como objetivo primordial la difusión y la promoción de la ciencia y la tecnología a nivel nacional y servir como consultor en ciencias a los organismos del Estado. Sus fines son promover el desarrollo de la ciencia y sus aplicaciones, conocimientos útiles para las necesidades de la sociedad peruana, fomentar la excelencia en la enseñanza de la ciencia y la investigación desde los primeros años escolares hasta los niveles superiores, estimular interacción entre científicos peruanos en el exterior y los residentes en Perú, así como con los de la comunidad científica internacional, apoyar proyectos de coordinación internacional con efectiva transferencia de información y conocimientos, incentivar la participación de mujeres en los programas de educación científica y actividades científicas a todo nivel, promover publicaciones relacionadas con ciencia, tecnología y desarrollo del Perú, alentar a los científicos de mayor nivel a publicar en revistas internacionales. Asimismo, la Academia Nacional de Ciencias del Perú fue aceptada en el 2003 como miembro científico del Consejo Internacional para la Ciencia, International Council for Science ICSU. Además, es miembro de la Red Interamericana de Academias de Ciencias, Inter-American Network of Academies of Science, IANAS. A través de los puntos focales peruanos designados en noviembre del 2012, participa en sus programas prioritarios: agua, energía, enseñanza de las ciencias y mujeres para la ciencia. Los puntos focales peruanos tienen a su cargo publicaciones sobre Perú, reuniones de IANAS, organización de seminarios y talleres. También se debe destacar la presencia actual de la Academia Nacional de Ciencias en la Academia de Ciencias del Tercer Mundo, TWAS. Los presidentes de la Academia Nacional de Ciencias desde sus inicios hasta el presente son Dr. Godofredo García, Dr. José Tola Pasquel, Ingeniero Alberto Guise, Dr. Roger Guerra García, Dr. Ronald Woodman, Dr. Abraham Weisberg, Dr. Gustavo González, Dr. Alberto Gago. Desde su fundación, la Academia tuvo un espacio en la Universidad Nacional Mayor de San Marcos. Luego, con el Dr. Tola Pasquel se mudó a otro en la Pontificia Universidad Católica del Perú. Recién en el 2001 el Ministerio de Educación nos cedió parte de las instalaciones de la Casa de Osambela, Centro Cultural Inca Garcilaso de la Vega, que actualmente es nuestra sede, así como de la Academia Peruana de la Lengua, Academia Nacional de Historia y Academia Nacional de Medicina, además de la Academia Peruana de Ingeniería y Academia Peruana de Derecho. La fundación de la Academia Nacional de Ciencias se debió a un grupo distinguido de peruanos dedicados a la ciencia, la mayoría profesores universitarios. Después de varios acuerdos preparatorios, el 6 de agosto de 1938, en el Salón de Actos de la Facultad de Ciencias Biológicas, Físicas y Matemáticas de la Universidad Nacional Mayor de San Marcos, se realizó la sesión de instalación de la...
Muy buenas tardes con todos, bienvenidos a este evento del día de hoy. Mi nombre es Ian Vázquez, soy el director de la Red Peruana de Ciclo de Vida y Ecología Industrial, y el día de hoy, en una colaboración con la Academia Nacional de Ciencia, estamos organizando este evento que se enmarca realmente en la semana grande del Grupo de Investigación Pelcan, ya que estamos de 20 aniversario. Entonces, como ustedes saben, el día de mañana pues tenemos un evento un poco más grande al que están todos invitados, pero hoy junto con la Academia Nacional de Ciencia pues tenemos esta charla, y para introducirla tenemos con nosotros al actual presidente de la Academia Nacional, el doctor Alberto Gago. Muchísimas gracias.
Gracias, Ian. Bueno, buenas tardes. Voy a hacer un breve speech en inglés. So, good afternoon, everyone. Welcome to this conference organized by the National Academy of Science of Peru in partnership with the Pelcan Group, the Career in Environmental and Sustainable Engineering, and the Science and Engineering faculty at PUCP. The National Academy of Science is a non-profit organization with more than 100 members. The members of the Academy come from a broad range of fields that go from exact and natural science to social science, including, of course, environmental science. And one of the main goals of the Academy is the dissemination of scientific knowledge, with special interest in disseminating topics related to to global concerns. Today, we are honored to have as a speaker Dr. Johann Fellner. He is an associate professor at Technische University of Vienna, and he is a world-class scientist in environmental science in topics such as waste management, circular economy, and anthropogenic resources. His work has reshaped the modern understanding of waste as a strategic resource. Today, he's going to talk us about waste characterization in the lab. Now, the floor is yours, Dr. Fellner.
Thank you very much. Thank you very much for the invitation and also the introduction. The title I was requested to talk about is From Sampling to Data Characterization of Waste in the Lab. And you will see later on during my presentation that I put the focus more to the sampling because I guess it's the most crucial part when we talk about waste characterization. So the content, I would like to introduce you to the topic. Then, as I said, talk a lot about sampling, and sampling is not only in the field but also in the laboratory, sample preparation, and then the final analysis. Typically, these are results what we obtained finally from the laboratory, or these are the results what we get after we did all the analysis. This is, for example, the resource potential of a certain type of waste, of incineration residues, of ashes from waste incineration. And what you see here is the content of different metals in different grain sizes, so in different fractions, starting from very high or large particles, going down to smaller particles, and it's a cumulative information. So this is what we get at the end, but how do we get to these results, and how we get in a very reliable way to these results? And this I would like to show you and guide you. Usually it starts here. Yeah, this is basically a heap of incineration ashes, and at the end of the day, we would like to know how much metals are in there with a certain reliability. That could be one starting point. Another starting point, as we see here, that's another analysis. What we did recently is construction and demolition waste. Simply, the picture already indicates a lot of challenges which are arising when we would like to characterize this type of waste. Or a third topic where we are also dealing with is the characterization of municipal solid waste, of mixed municipal solid waste, and like how much valuable plastics is in there. And the question is the same, the procedure, the sampling is the same, but we need to consider different aspects. So what are the challenges when we talk about sampling of waste? And I believe the biggest error in waste characterization is typically done when we talk about sampling. So the first, one of the big challenges, we have large quantities which we need to characterize. So again, here ashes or mixed municipal solid waste. So basically, Thank you. So we have large quantities we need to characterize, and at the end of the day we need to get representative samples which needs to be analyzed. So this is one big challenge. Another challenge that we have a mixture of very different materials. Okay, thank you. Yeah, as I started, huge quantities, mixture of materials which behave very different, which have very different properties. Another challenge is the big variation in particle size. If you have a lot or population which has a similar grain size, it's much easier than if you have very large particles and very small particles. That's another challenge we need to consider. What we experience also in the field, but also in the laboratory when we do sampling, is very often because of these diverse characteristics, we have a kind of segregation of materials. What is shown here in this rather brown red color is roasty iron particles in the waste, and it becomes obvious that at the end of this heap here we have higher concentration of these materials than within the heap, simply because of they behave differently. And the same applies when we talk about plastic wastes. Bulky materials tend more to be at the bottom, whereas lighter materials like plastic films tend to be more accumulated somewhere at the top of this heap. So all this is challenging for sampling, and sometimes we have the target material present, the analyte present only in very small amounts. This was a study we did in Uganda, where we would like to characterize the quality of the compost they are producing, and we would like to know the pollutants or the quantity of materials which should not be in there, like these needles here. And in order to get reliable information about how much is in there, it's also challenging because, as mentioned, these materials are present in very tiny quantities. And finally, this brings me to the last slide for the challenges of sampling. We start with a very large population. In that case, maybe 200 tons of materials, which should be characterized for, I don't know, the metal contents or whatever. Then basically we are taking about 200 kilograms out of the big lot into the laboratory. This is our field sample. Prepare it then in the laboratory, and finally, and this is really the big challenge, finally for the analytical device in the lab, we can only analyze typically about a few grams or less than a few grams even. Sometimes it's only about 50 to 200 milligrams, which is analyzed. But what does it mean? That this small quantity needs to be representative for the whole lot here. And this is the big challenge, challenge with regards to sampling, but also challenges with regards to the preparation of these samples. And how we can approach this topic in a scientific way, I would like to show you now. A non-scientific way would be simply that we go out there and try randomly to grab samples. This is unfortunately how it's done very often. Very often people put a lot of effort in the final step in the analytical device in the laboratory, but good sampling is very often disregarded because simply grabbing a sample is very fast. We don't need a lot of staff members, we don't need special equipment, and we have a sample, and that's fine. But basically this should not be done this way. We really need to do a good sampling plan of how to take samples out of the waste and how to prepare these samples. That brings me now to the sampling, or the correct sampling, and there is even a theory out there of sampling, waste sampling, which has been not developed for waste but for mining material, and more or less the pope of waste sampling is this guy here, Dr. Pierre Xi, a French researcher who wrote a lot of books on sampling, on the theory of sampling. He died about 10 years ago, and everything which relates to sampling is basically based on his theoretical work he developed. I mentioned already what are the challenges, but we can try to put that now also in a more scientific way. The major challenge of waste sampling is the heterogeneity of the waste. Yeah? We don't have a homogeneous material, we have a heterogeneous material, and based on the theory of sampling from Mr. G, we are distinguishing between the so-called constitutional heterogeneity, I'll explain later what is that, and the so-called distributional heterogeneity. Both of them play a major role when it comes to sampling. You see now here a picture of what is meant by the constitutional heterogeneity. Imagine this is the population, and the black bubbles here represent the analyte of interest, what we would like to know. And we take now sample A and sample B, yeah? In order to get the representative information of how much of this analyte is present. It becomes very obvious that sample A is basically not a good sample because there is no analyte present. We don't have any clue whether sample B is already representative, but at least we have some of the analytes in there. And the constitutional heterogeneity basically describes somehow the heterogeneity with regards to the analyte. If the analyte is present in a large quantity or in a high concentration, the sample size can be definitely much smaller than if the analyte is only present in a very few, in a very low concentration. Later on, I will show you a practical example. What does it mean exactly? So when we talk about this constitutional heterogeneity, it is connected to the so-called fundamental sampling area. On the other hand, we have the distributional heterogeneity, meaning that the analyte we are interested in is not equally distributed or randomly distributed. We have certain areas in our population where we have a high concentration of the analyte, and maybe some areas where we don't have any analyte in there. And this error is the so-called grouping or segregation area, or sometimes people talk also about the minimum practical error. So these are the two errors we need to deal with when we talk about sampling, and the idea is that we keep these errors as low as possible. I have here a small example which should highlight again this constitutional heterogeneity. It's not a waste example, it's a food example. Imagine you're taking your breakfast and you have your muesli, and you would like to be interested what is the content of raisins in your muesli, and how big should be the sample you are taking in order to get the representative information about the raisins content in your muesli. I did that for you now. I took theoretically samples. That's the first sample, second sample, third sample, fourth sample, and the fifth sample. Okay? And now we can try to quantify simply because of the area covered by the raisins, what is the raisin content in that sample. For sample 1, I said it's about 14%, sample 2, 21%, sample 4, 42%, in sample 3 we almost don't have raisins in there, and sample 5 is 25%. So we take samples with very different information, and if we take a sample size which is big enough, the result of each sample should be the same, almost the same. In our case, we could quantify an average or a mean value of about 21%, a standard deviation of these four values which is 14%, and when we refer the 14% to the mean value, we have a relative standard deviation of 69%, which is rather big. And this 69% relative standard deviation is the so-called fundamental sampling error. Okay? Because if we would lower or eliminate the fundamental sampling error, that means each of the samples should give more or less the same quantity of analyte. So when you just see these pictures, what can we do, or what do you think we can do in order to reduce the fundamental sampling error? It's rather easy. What would you suggest? Any idea? We have these 5 samples taken here. What would you recommend in order to have a lower fundamental sampling error? We simply would need to increase the sample size for each of them, because if we increase the sample size, the possibility that we have the same number or the same quantity of raisins in there would rise. So in order to tackle the fundamental sampling error, we need to increase the quantity. And Piaget developed some theoretical formula to calculate the fundamental sampling area. I don't want to explain that in detail, but what he found out that the fundamental sampling area is very much based on the sample mass, on the grain size, or the particle size of each of the particles which are in there, on what is the mean content of the substance to be determined, and what is the content of the so-called target materials or carrier particles. Yeah. If we go back again to the previous picture, increasing the sample size would simply mean that the picture will become bigger, and the possibility that raisins are present in a more uniform way is higher. So that definitely makes sense. Another point what he mentioned is if the particle size gets smaller. So imagine if each of the particles which are present in the picture is much smaller than the ones which are given here, then we definitely get also a more equalized picture for each sample. Yeah? So the particle size is quite important. And of course also how often this particle arises. Yeah. Based on this theory, he developed this formula which allows us to calculate what is the minimum sample mass we need to take. Yeah? And this formula is given, that's the quantity of sample we need in kilograms. Then it's 1 over omega. And omega is the expected mass fraction of analyte-bearing components. So we need to have already some guess what might be the concentration. Yeah? So you can only apply this formula if you have already some idea about what we expect. Typically, we would use this factor from previous studies which have dealt with similar type of wastes. Then the formula contains the desired relative standard deviation. This is like the relative sampling area we are allowing. Yeah. So this is the sampling area we how to say accept. And then finally, we need to have also information about the grain size of the particles inside the lot, inside the population. On the one hand, we need to have an assessment about what is the weight of the heaviest particles in there, and also what is the typical mass or the typical weight of the smaller particles, so the 10% parts centile particles. So applying this, we can assess what is the right quantity, or at least the order of magnitude of the sample we're supposed to take. There is one thumb rule when we apply sampling. We should find in our sample mass, what we take, about 100 particles we are interested in. Imagine you would like to know what is the quantities or what is the concentration of batteries in our municipal solid waste. Then the sample size, the correct sample size would be that we find at least 100 batteries inside the sample. If we do so, our final sampling error will be about 10%. If we, if we accept a higher sampling error of 20%, that means we are fine already with 50 particles or 50 batteries in there. If we would like to know the battery concentration with a very high accuracy, with a relative error of about only 1%, we would need to take a sample which is really very big, because we need to find 10,000 batteries or 10,000 particles of interest in there. So it's the relative error we are accepting determines to a large extent the sample size. This is a picture which should highlight that sometimes we are facing also challenges with the constitutional heterogeneity in extreme. This is the so-called nugget effect. Again, our muesli, which contains only a very few target materials. That means even if we take a large sample out of it, very often our raisin concentration, which we would determine, is very low, or even zero in that case. And this is something we observe very much in waste analysis. This is a study which we have done longer time ago, where we try to measure the copper concentration in a certain type of waste. And we see, we take about 16 samples, and most of the samples show a copper concentration close to zero. Yeah? Only a few samples had a higher concentration of copper. Very often people tend to say this is an outlier and neglect the high concentration values. But this outlier should not be, neglected simply because of this nugget effect, meaning we do have some parts in there which might contain copper, but the big part doesn't contain copper, and if we neglect the few samples where we detect copper, then we underestimate the real concentration of copper in there. That was the fundamental sampling area. Now we come to the distributional error or segregation error. Again, our muesli, and now it's not randomly distributed. We have some areas of high concentration of raisins and some areas where we don't have raisins in there. What we can do in that case, it's not recommended here to increase the sample size. What is more recommended here is that we take small increments, take smaller samples from different places of our lot, and finally produce one subsample in order to have distribution or samples taken, subsamples taken from different sections in order to represent the whole lot. So distributional heterogeneity can be covered by having a lot of increments. Another option in order to improve the sampling here would be to mix the lot, but very often this is not feasible, so we need to try to take samples from different places. The number of increments we take helps us to reduce the segregation error, and typically it reduces the segregation error by the square root of the number of increments. So meaning when we take 4 increments from different places, we can reduce the segregation error by factor 2. If we take 100 increments, we can reduce the segregation error by factor 10. this is now a practical example what could happen. Imagine we have materials look like this, and we would like to get an average information about the quantities of those stones here or whatever it is, yeah. And we have a sampling device which looks like this. Or in the other option, we have a sampling device which is rectangular. This one is circular. Which one of the two you think is better to take samples? The upper or the lower one? Which one delivers more representative samples? Who is for the upper one? Who is for the lower one? Okay, the majority. Okay, thanks. You are right. Because if we take the upper one, that means we are having a lower representation of the particles which are at the lower end here, and we see a higher concentration of these materials at the lower end here. That means only if we take this rectangular sampling device, we are taking samples in the correct manner. Finally, when we talk about sampling, there is one statement which highlights what is really important. Whatever is the distribution of the materials, the sampling or the selection is only correct if all these elements of the volume have the equal probability to be selected. And as we saw here in the example before, the materials which are at the lower end here have, using this sample device, a lower probability to be selected than the upper particles. Yeah? Whereas here, over the whole depth, we have the same probability that the materials are selected. This brings me to another challenge. Very often we have lots, populations like this, like a big heap. This is a three-dimensional lot. This is a two-dimensional lot. This one is waste on a conveyor belt. When you need to decide where to take samples, which of the three different lots would you prefer? Where do you think it's easier to take samples and where we have the highest probability that each of the particle has the same probability to end up in the sample? Is it this big heap where we have, I don't know, 5 meters or 3 meters of waste? Is it this two-dimensional lot, or is it here on the conveyor belt? Any idea? Yeah. Number 2? Okay. You are for the last one? Yeah, okay. This one is the most preferred option because here, of course, it becomes very obvious that if we have a big heap, the probability that we are digging into the heap to take out samples is very low. So typically people just take it from the surface. Yeah. So this is definitely the worst case. Here we have the big risk. I mean, this is a rather small lot, but if it's over a bigger area, the possibility that we take materials from the middle is also lower than from the side. So this is again not, it's of course more preferable than the three-dimensional lot, but here it's has the highest probability that we are taking materials with equal probability from different places. So this is the most preferred option where we're supposed to take samples, and sometimes we even try to bring a three-dimensional lot into a one-dimensional, like here. So this heap is then distributed over a long distance, and then I don't know if you can see it here. Then people can take cross sections at different places that allows us to take good samples. Another possibility is, if we have a conveyor belt that's most convenient, that if we bring the three-dimensional lot over a conveyor belt, and at a certain time point at the end of the conveyor belt we can try to take samples, which is maybe most convenient. Now we took the samples, and we are going into the lab, and the question is now, we have about, I don't know, a few hundred kilograms of samples taken from the field, how we can finally derive the quantity, a representative quantity we need for the analytical device. So it basically starts, it basically starts with the sample, the field sample. Then we need to do milling and fractioning. Milling means we simply reduce the grain size. By reducing the grain size, we are also reducing the constitutional heterogeneity. After we have reduced the grain size, we can reduce the sample mass, so we can divide the sample and reduce it, and then have a secondary sample, and then the steps are repeated. So we have again a milling, sometimes fractioning, so taking out particles which are hard to mill in order to reduce the heterogeneity. And then finally we derive with the analysis sample. These are typical devices we can use in the lab to reduce the grain size. This is a cutting mill, which allows us to have a size reduction to a size of about 1 to 4 millimeters. These are then further milling steps. Ultracentrifugal mill, which allows us to reduce the grain size to about 0.2 millimeter. And this is a cryo mill, which is cooled down, especially for milling plastics, which allows us to have a size reduction lower than 0.5 millimeter. The following numbers should give you an idea how important is the reduction of the grain size. Yeah? If we have a sample of 1 gram with the average grain size of about 2 millimeters, so somewhere in between, we have only about 200 to 1000 particles inside the sample. For 1 gram. As I said, usually we just can put about 1 gram or half, or sometimes even less than that, 500 milligrams into the analytic device. It's quite important to reduce the grain size further to that low size, because only in that case we have a high quantity of particles in there, and the higher the quantity of particles is in there, the more homogeneous the analyte is distributed, because I told you we would need, for an error of about 10%, we would need at least 100 target particles in there. Yeah? So this requires us to reduce the grain size a lot. Fractioning is done for those particles which are hard to mill. For example, metal pieces are taken out separately. So what is applied there, we are usually trying to sieve the samples and then very often manually sort out the materials into like I think that was plastics which have been removed, or glass. And these steps can be used in addition to milling for those materials which can be separated manually. For the mass reduction in the laboratory, we use either riffle splitters, where we feed in the material and then it's separated, or sometimes rotation splitters. So the samples are fed in here and then these bottles are rotating. And the whole sample mass is then divided into typically 6 to 8 bottles, so we reduce the sample size by factor 6. Or very simple separation is incremental samples, so we have the sample and then we split it into, in that case, 4 subsamples and reduce the sample mass by 4. And then we take one of the subsamples and then go again into milling or into fractioning and repeat this step several times in order to come from the few hundred kilograms down to the 1 gram or 0.2 gram we need. Here is an example which also highlights how important is the correct milling or the grain size reduction. This is an analysis which is done for refuse-derived fuels. So fuels which are derived from waste. And the idea here was to analyze the content of carbon, hydrogen, nitrogen, and sulfur, because they are very important for the combustion characteristics of these types of waste. And what we did there was we investigated what has the effect of the sample preparation, so the milling, on the final results. And we used 2 different types of mills, the Cryo mill and the ultracentrifugal mill. And we used exactly the same type of RDF. And what we found out that this type of mill was much more preferable because all the samples had a lower variation at the end when we analyzed it with regards to the carbon content and the hydrogen content. Whereas the other mill, which finally produced materials of a higher grain size, was less favorable because we had a much higher variation. If milling and sampling would be perfect, we would have a very low variation of the different samples. Another device which is used for waste characterization, or which we very often use, is ICP-OS, which can help us determine the heavy metal concentration in wastes. So typically first the material is dissolved in acids and then it's inserted into the device and heated up at very high temperatures, plasma of 10,000 Kelvin, and there the emission lines are detected, and out of it we can determine the concentration of the different heavy metals. And this is then finally the result we can get out of such analysis. So that was the first slide I was shown, and that's also the last slide before I come to the summary. I believe that sampling is very often underestimated in waste management, but it's very crucial for good results. And it's very challenging, especially in waste management, because we have typically a very heterogeneous material, making it difficult to get representative samples. And we can have the best analytical device in the laboratory. The errors what we made during sampling can never be corrected. This is something quite important. Sampling is very time consuming. It needs a lot of effort, a lot of money also. And yeah, we need to have enough sample size and enough increments to capture the heterogeneity. And what I also recommend, that if we have a sample in 3 or 2 dimension, that we try to convert it into 1-dimensional lot, because then sampling gets more easy and it's more representative. Yeah, and finally in the lab we also have sampling steps, because we need to reduce the grain size in order to finally analyze it, and every size reduction is also connected to sampling, and this is quite important to be considered. Okay, that's from my side. I think I kept the time almost, so thank you very much.
Thank you very much, Johan. Really helpful for everyone that is doing this type of work, not only in waste management, but in general, everything that is related to understanding what happens in the environment and using the correct tools in the lab. So we have time for some questions. Who would like to ask the first question? Yes, Luis, please wait for me.
Thank you, Professor Fellner, for your questions and for your presentation. I have a question in order for your sample. At the beginning, you mentioned that you have to ensure for your lab distribution and heterogeneity, right? So how you can ensure this at the beginning of the process? I mean, when you collect the big sample, for example, this kind of tons of waste, how you can ensure this kind of representative of the city? You have like restraints of the system, like you determine like a temporal or some territorial limits.
Thank you, Luis. Thank you, Johan.
Very good question. So I guess you are asking about how to be sure that, for example, for a city like Lima or any other city, you are getting representative samples of your municipal solid waste you would like to characterize. Yeah. In that case, from the very beginning, we would try to lower the heterogeneity of the different samples in a way that we try, first of all, to separate the city into the different districts with different socio-economic conditions, because waste in the poorer area will be definitely very different to the waste in the richer area. So that would be the first step, to take samples from different regions or from different districts. You mentioned that also the temporal situation might be different. I don't know, wastes on Mondays might look different than wastes on Fridays. So also this is supposed to be considered, that you are trying to take samples during different days of the week, and not only different days of the week, but also during different seasons. Yeah. I think that doesn't play a big role here, but we had some sampling campaigns now in Ghana, and they have very different seasons, with wet season and dry season. So it was definitely necessary to take waste samples during the wet season, which has a much higher water content, because the waste containers there are opened, not closed, meaning it has a very big impact on this. Yeah.
Thank you, Johan. Yes, we have a question here from Alejandro.
Thank you very much for the presentation. Very nice. I have one comment and then one question. While you were presenting, I was just thinking on this new boom on the rare earth metals or elements. And a lot of companies now are putting the eye on mining waste. So because you want to extract them from mining waste where these elements are more concentrated. So this applies super well, I think, to this new niche. And my question has to do with organic waste management. From my experience with black soldier fly farming, some producers get the waste from municipalities, and then there is machinery that can separate the plastic from the organic stream, and then usually the organic stream is mixed with, is shredded and mixed sometimes with water, right? So I guess, I don't know if you have any insights on organic waste. This shredding already makes the work of mixing, right? Thank you.
The shredding, on the one hand, mixes the waste, so it reduces this distributional heterogeneity, and on the other hand, it reduces the grain size, and the smaller the grain size gets, the lower the minimum sample mass gets, because it's you have smaller particles in there and they are more equally distributed. Yeah. What do you mean? Yeah. With slurry, okay. So yeah. So I would not recommend, for example, if you have a container and there is the container is filled, I would not recommend to take the sample out of the container, because it's getting difficult. That means we would need to take samples from the container in different depths. If you have a sampling device which allows you to dig down in the container to take also samples from the bottom and not only from the top, because the water content at the bottom might be much higher. Yeah. So that makes it rather difficult. So if it's possible, somewhere in the processing way, I would recommend to find another point of sampling. Yeah. But if you need to take it from the container, it is really necessary to try to take samples from different depths and different places in the container. But in practice, very often what's happening is that people take the easy way and take samples from the top of the container. Yeah, but this is incorrect.
There's one question from Ruben.
Thanks for your presentation. I think your presentation was focused mainly on uh, um, huge examples, right? But I have a question, maybe on a parallel system, which is a million times smaller, which are microplastics. Right now, we have not only a sampling procedure, but some kind of procedure that is time consuming in order to manually detect the fibers, fragments, microbits in a sample. So my question is, what considerations can you suggest in order to maybe um, um, to improve this kind of sampling in order to reduce the time right now we have on this procedure?
very difficult question. What is the media you are taking samples from the microplastic? Is it water samples? Yeah, so you're taking. Sorry. Okay. Very low concentration. So the first step is that you are filtering out the plastics. Yeah.
Yeah, well, we are filtering, and then we have some preparation of the sample using some chemicals to remove organic matter.
Okay.
And then we isolate that fibers or microplastics, filtering them.
Mm-hmm. I mean, um, I would say the rather good thing is that within the water body, I believe that you have a rather good mixing of the different materials. So how to say, the microplastics most likely will not be concentrated in certain locations in the water. So this first step is maybe easier. For the second step, I don't have really any recommendation because you have so tiny concentrations. One question would be, what is the concentration of microplastics in comparison to other solids in there? I think even this is very small, yeah? Yeah. So, most of the studies, I think today are more or less on a, I would say, semi-quantitative way when it comes to microplastics. I don't see any serious. I mean, maybe someone could try to apply this formula also to microplastics and to the solids, but we need to have at least some root information about what is the particle size and what is the concentration of the other solids in there. Yeah, and then we could try to calculate what is the minimum quantity of solids which have been filtered out we need to take. Yeah. I think the principle of the formula and the principle of the sampling theory should be the same, but we need to put that number in there first, or some best guess, in order to see how big should be the sample. Yeah. Sorry. Any further question?
Yeah, well, thank you for a great presentation. My question is a bit more practical. So I'm thinking more about, let's say, a small municipality in which they probably don't have access to all this great information, no? So on the one hand, which would be like the 3 or 4 basic principles they should consider to at least aim to do a decent characterization of the waste? And the second part of the question would be, what agency could they rely on maybe to find some basic information on the internet or whatever, just to have like some minimum pillars they can follow and have kind of a short induction to get the minimum information?
If you don't want to follow the theory and the formula, there are some standards out there of how to do waste characterization for mixed municipal solid waste. I don't know the exact abbreviation of the. It's an American standard, AMTS. I can send it to you. So there are guidelines out there depending on the size of the or the quantity of waste, which procedure they should follow. Yeah. So this was how to say, the theory behind that, and the theory has been put into practice in that standard. Yeah.
Great, Johan. Thank you very much. I appreciate your time and bringing the theory with your more than 20 years of experience in this field. So as you know, Johan has been working on waste management for more than 20 years, 25 years actually, and in TU Vienna, doing really fantastic work on this subject. Not only waste management, but as you can see, also everything related to incineration of waste and the other fields, and also things related to urban stocks. So again, thank you very much for coming, and it has been our pleasure to have you here. Thank you. Vamos a cerrar la presentación hoy día. Muchas gracias por asistir. Y bueno, también Johan se va a quedar unos minutos si quieren hacer alguna pregunta en español que podríamos traducir también en caso lo prefieran. Gracias.